thank you all for joining us today for this webinar. We really appreciate you taking the time out of your schedule to learn about Tenaya, our programs, and the science behind them. My name is Wendy Borsari, and I'm the Associate Director of Patient Advocacy, excuse me, at Tenaya Therapeutics, and also a patient with hypertrophic cardiomyopathy myself. I want to just go over a few pieces of housekeeping before we get started. So there has been one change in tonight's agenda. So Dr. Laura Robertson, who is going to talk about our MYBPC3 HCM program, was unable to join us this evening. So Dr. Matt Pollman will be talking about both of our programs tonight, our HCM program and our ARVC program. You'll see at the bottom of your screen that there is a Q&A box, so please feel free to drop questions in there at any time during the presentation. We will answer questions at the end, but drop them in there while you're thinking of them. Due to time constraints and the inability to answer some questions that you may pose because the information is not yet publicly available or publicly known, we may not get to all of your questions today. But at the end of the webinar, we will share an email address with you. And if you have any questions that you would like to follow up on, any questions that you think about later, or other questions that we just didn't get time to answer, please feel free to send them to us, and we will do our very best to answer them for you. Your questions are very important, and we want to make sure that we do our best to answer them. Please note that the raise hand feature and the chat feature will not be available for you to input information into. And that is really to protect your privacy. We don't want to see identifying information. So those things have been turned off for this. A recording of the webinar with the slides will be available afterwards on our YouTube channel. So we will be sure to let people know when that is posted online. So it might take a little bit of time for us to get it up there, but we will have a video available for you in the future. And now I would like to introduce our first speaker, Dr. Whit Tingley, who is our Chief Medical Officer. Oh, yep, I'm sorry, I forgot to go through the agenda. So this is our agenda. So we're going to introduce you to Tenaya. That's what Whit's going to take care of. The basics of gene therapy. And then Dr. Pollman will take you through the MyPeak and Ridge trial. And then one of our scientists, Dr. Laura Lombardi, will take you through the early science. So if you can just go to the next slide for our forward-looking statement. And then I will pass the baton to Whit. Thank you so much.
Thank you, Wendy. And thanks everyone for joining today, for your interest in gene therapies for genetic forms of heart disease. Next slide, please. Here at Tenaya, we are fully committed to improving options for patients with inherited cardiomyopathy. Tenaya was formed in 2016, and we've been 100% focused on heart disease from the beginning, and with a particular focus on genetic heart diseases. We currently have 5 clinical trials related to genetic cardiomyopathies, including 2 active gene therapy trials. One called MyPeak-1 is, as Wendy mentioned, for hypertrophic cardiomyopathy due to mutations in the gene called MYBPC3. The other trial, Ridge-1, is for arrhythmogenic right ventricular cardiomyopathy due to the gene PKP2. These trials have been designed with patient input and patient needs in mind. We very much want to hear from patients, from you. We have a dedicated advocacy team, including Wendy, and an advisory council of patients that gives us feedback as we go through the development process. We also strive to help educate patients, families, and everyone more broadly, including events like this one today. And you can find more information on ARVCstudies.com and HCMstudies.com. We have many values at Tenaya, but our first and foremost value is patients first. Our mission is to transform and extend lives through discovery, development, and delivery of potentially curative therapies that address the underlying causes of disease. And for genetic disease, that means gene therapies. Many opportunities for patients, families, friends to get involved in drug development to help advance the next breakthroughs in treatments for diseases. And the cardiomyopathy community has a key role every step of the way. Here at Tenaya, even when we start in preclinical research, we want input, understanding the impact that the disease has had on you, what you see as the unmet needs, and what you need next. We also more formally collect data through natural history studies where patients volunteer to share their information over time so we can understand exactly what happens over time and what the needs are. And as I mentioned, we have a very active patient advisory council as well as individual focus group events. Participate with us, participate with others. It's extremely helpful. Now, during the clinical development is divided into different phases, 1, 2, and 3. We don't have time to go, and 4. We don't have time to go into the specifics of the phases now. Happy to answer questions later if you have them. But in addition to directly participating in trials, helping other individuals and families decide about whether participating is right for them. Reviewing clinical trial designs with sponsors. How is this friendly towards patients? What could be improved? Informed consent forms, are they understandable? Will most patients, get the important points from this informed consent form? Also talking with regulators, the FDA is very interested in direct feedback from patients about their experiences, what their needs are, and what they hope for from therapies. So please participate in FDA-sponsored patient events. Next slide, please. So we're focused today on 2 genetic diseases, hypertrophic cardiomyopathy due to the MYBPC3 gene and ARVC due to the PKP2 gene. Both share the themes shown on this slide. So a normal functional gene produces a protein, and it's the protein that carries out the key cellular functions that help the heart work. In both the cases we're talking about today, the gene has a misspelling in it, which causes the protein not to be produced. So patients do not have enough of a particular protein, and that disrupts a particular function of the cell, the contractile function in the case of HCM, and the electrical function in the case of ARVC. And so the loss of these proteins result in cardiomyopathies. And then once you have the cardiomyopathy, there are a variety of other downstream effects. So that can lead to heart failure in both cases. So we realize that many patients don't have easy, convenient, or cost-effective ways to get genetic testing to determine if they have a genetic disease and what the gene is. So we sponsor a free genetic testing program that includes genetic counseling as part of the program. And this is for adults with suspected or diagnosed hypertrophic cardiomyopathy or ARVC. There's no obligation if you participate in the trial, certainly no obligation to participate in studies. So to learn more, please directly go to our patient advocacy group at patientadvocacy@tenayatherapy.com, or your physician can contact our medical affairs group through the medaffairs@tenayatherapy.com email. So gene therapy for HCM and ARVC are investigational. We're still doing trials in patients to understand the safety and efficacy of the therapy, but the goal is to treat the underlying genetic cause of the disease and ultimately to halt progression and potentially reverse disease after it has started. We are targeting the specific underlying gene and replacing the broken copy with a new fully functional copy. And that will restore the normal protein levels and help the heart cell function to recover. We hope in clinical trials to demonstrate meaningful improvements in heart rhythm, heart strength and function, heart relaxation, and most importantly, patients' symptoms, quality of life, and prognosis. We have data from animal models to support all of these, which we will talk about later in this session. These gene therapies are administered once, a single-time dose that's given as an IV infusion, and we'll get into more detail about how they work, but once they're infused and get to the heart, they have long-lasting effects. We do not know yet in patients how long it will last, but the expectation is that this will be measured in years. These gene therapies do not modify egg or sperm cells, so it does not affect patients' children. The children can still inherit the disease. They do not inherit the therapy. So this shows more detail about how our gene therapies work. The goal is to deliver the fully functional gene to the heart cells, and so we need a delivery system. And that polygon there is the AAV viral vector. So you'll hear it called vector, you could hear it called capsid. How I think of it is this is the FedEx box. So it has the address information to bring the package to the right part of the body, in this case, the heart, and it protects the contents during the delivery process. But once it's delivered, it doesn't matter. The viral capsid is broken down. The best delivery technology that exists today is based on pre-existing viruses, and in particular, these adeno-associated virus, or AAV. These occur naturally. They don't cause disease in people, but they do infect people and do get to various tissues in the body, including the heart. What we do is we use cells to make new viral packages, but without any of the viral contents. That it can't replicate, it can't do an infection in a typical viral sense. We just use that package for the delivery, and we replace the contents with the working gene, so MYBPC3 or PKP2. As I said, this is a single IV infusion. It goes around the body and then hones to the cardiac myocytes. It does go to other cell types as well, but there are specific factors in here to cause the gene to only turn on in heart cells and cardiac myocytes. So we restore normal protein levels and restore the function of the heart after the gene therapy. That is the vision. Again, please put your questions in. We will answer them at the end of the presentation. There are potential risks to gene therapy. We are fortunate that AAV therapies have been studied in many trials, and in many cases, we have approved therapies. So we understand a lot how the body interacts with AAV-based gene therapies. And what we watch out for most is the immune system's reaction. The immune system doesn't know this is a therapy. It assumes it's a virus that it should get rid of. So it can potentially get rid of the therapy so that the therapy wouldn't work. The reaction can also cause inflammation, and in particular, inflammation in the liver has been observed. So we use specific medications to suppress the immune system's reaction to the virus. So anti-inflammatory medicines to prevent that reaction, allow the gene therapy to work. At this point, with AAV-based gene therapies, patients can only get treated once. So when the immune system has seen this virus and had a reaction, it develops antibodies that will then clear the next time that gene therapy is dosed. So it is truly a one-time therapy. As I mentioned, the viruses do go to other cell types, so it can deliver the package to the incorrect cells. We are fortunate in the case of the heart that there are very clear switches, we call them promoters, that turn on the gene only in heart cells. But this is still a theoretical risk of our gene therapies. And of course, our therapies are still under investigation. We are in clinical trials, and not all of the risks are known today. There have been substantial successes for patients using AAV-based gene therapy. The FDA has approved 5 for eye disease, for muscle disease, for blood disease, and for neurologic disease. And similarly, the EMA has approved many of these same medications and also gene therapies for metabolic disease. So really great successes for these patients, and our hope is that soon cardiac patients will also benefit from AAV gene therapies. All right, as Wendy said, Dr. Robertson could not be available today, so I'm happy to introduce Dr. Matt Pollman to present the specifics of our 2 gene therapies and the clinical trials.
Great, thank you, Dr. Tingley. It's my privilege to be able to walk through at a high level the 2 trials, one for the hypertrophic cardiomyopathy and the other for the arrhythmogenic right ventricular cardiomyopathy. So we'll start with the TN-201 MyPeak-1 as the name of the trial. And as Whit explained to you, the fundamental issue that drives the disease, the hypertrophic cardiomyopathy, is a defective gene which produces a broken, as Dr. Tingley termed it, a broken protein. So the goal here is, he explained, is to be able to reintroduce a normal copy of the gene that produces a normal protein and then supplements what is already existing. And the reason why we are pursuing this disease state is because it is significant. It's a significant cause of genetic heart disease, and is estimated to affect about 115,000 patients just in the United States alone, and many more worldwide. And as the name does imply, hypertrophic, it is causes a thick left ventricle. This thickness causes the heart to pump inefficiently, fill inefficiently, and sometimes there actually is obstruction as the ventricle tries to pump the blood out to the rest of the body. So this can lead to signs and symptoms of not receiving enough blood throughout the body in an efficient manner, and patients can be short of breath. They may have fainting spells, chest pain, fatigue, palpitations, and serious arrhythmias. So this is not a trivial disease. It does affect many patients in many different ways, and can lead to decreases in their ability to function, even the daily activities. Certainly, there are social and psychological impacts. There is an increased chance of cardiac death or heart failure. And we know that the typical age of onset is 40 years and older. However, younger patients can be affected. And when they're affected at a younger age, they often have a higher risk of severe disease with morbidity and mortality. Next slide, please. So although we don't have time, we're packing a lot into this one session to go through all of the criteria to be eligible for this trial, but this trial is enrolling now. This is our first in human clinical trial for individuals that are affected by the MYBPC3 mutation and have developed hypertrophic cardiomyopathy. In this case, we're first starting out with adults age 18 to 65. This will be what they term the non-obstructive hypertrophic cardiomyopathy due to this mutation. Certainly, individuals should be symptomatic, so we understand that the disease has progressed to a point in which there are symptoms and signs of the disease. For this trial and this early trial, we ask that the individuals have a functioning ICD. And of course, as Dr. Tingley explained, it's important to have the low neutralizing antibodies to be sure that the therapy can be effective as possible. The goals, particularly of early stage trials, are to assess the safety. That's job number one, and to compare a couple of different doses to understand what is the safest and most effective dose, and to explore as well the signs and the effect on the signs and the symptoms of the disease state itself. Next slide. So we try to be mindful as you look at the map of the clinical trial sites, again, to emphasize this trial is enrolling right now. And we've been mindful about the geographical distribution. to make it the less burdensome as possible for patients to be able to go to a site that hopefully is within at least driving distance, if at all possible. And there on the right panel, you can see all of the sites with the distribution: Oregon, California, Utah, Texas, Minnesota, Ohio, Pennsylvania, Massachusetts, and Georgia. And we've also provided resources for you. And of course, there's opportunities after the sessions to be able to explore these other resources to be sure that you can understand where the sites are and how to contact them. Next slide. So we certainly are in the process of dosing patients, and we announced the first patient dose back in October of 23. This was at the Cleveland Clinic. So again, this is very real and is happening now. Next slide. So I'll transition a bit to our other disease state that we're pursuing, again, caused by a defective gene. In this case, it's PKP2. This defective mutation causes a defective protein, and again, is the fundamental cause of the disease. It also is a severe and progressive genetic heart disease, and it's estimated to affect about 70,000 patients in the United States alone. Unlike primarily in the MYBPC3 HCM situation, this is less about affecting directly the heart muscle and the filling and emptying of the heart. This is more about the electrical conductivity of the heart. This is often underdiagnosed, and the first time, unfortunately, a sudden cardiac death. So the symptoms, as you can imagine, are more related to the abnormal heart rhythms, and they include palpitations or feeling irregular heartbeats, lightheadedness, fainting. And unlike the MYBPC3 HCM, this is actually a disease that presents in younger individuals, normally before the age of 40. So it does have a significant impact on the quality of life due to these arrhythmias. And if they do have an ICD in place, there certainly is anxiety about when that ICD may need to deliver therapy, which can be as aggressive as shocks. And certainly there are as well restrictions on physical exertion, which can exacerbate the disease in individuals. Next slide. So again, a high-level look at our first in-human clinical trial for individuals with the PKP2 mutation, the RIDGE-1 trial. Again, it's similar to the prior trial. This is for adults, need to have the appropriate mutation, and need to have evidence of disease. And in this case, is because it is a disease of arrhythmias and causes arrhythmias, we know that risk factors for arrhythmias are premature ventricular contractions or skipped heartbeats. And so there needs to be at least 500 of those per day, measured by standard measurements. Individuals should have an implantable cardio defibrillator. And again, just like the prior trial, low AAV9 neutralizing antibodies, with the similar goals to assess the safety of the 401 gene therapy, to compare the different doses to determine the safest and most efficacious dose, and to explore its effects on the signs and symptoms of the disease. So I will pause there, and I think the next slide, we will transition. One more slide. Just to give you a sense as well, the geographic distribution, similar to the MyPeak-1 trial, where we tried to be geographical mindful to make it as less burdensome as possible and the least burdensome to individuals. And we have sites in California, Colorado, Minnesota, Ohio, Maryland, New York, and Massachusetts. So again, there are resources for you to be able to explore, and we hope that you will utilize those. So next slide. And I will turn the presentation now over to Dr. Laura Lombardi, our expert in the preclinical studies.
Hello, thanks so much for joining us. I'm Laura Lombardi. I'm the Director of Gene Therapy Biology here at Tenaya. And today I have the pleasure of discussing with you some of the foundational science behind the clinical trials that my colleagues have discussed with you. So at Tenaya, drug development represents an intersection of myriad capabilities. I'll just highlight a few for our discussion today. At the core are genetic insights, and then using disease models in combination with AAV capsid engineering, as well as Dr. Tingley mentioned, optimizing promoter and regulatory elements for specific expression. And then, of course, optimizing drug delivery. We work to generate the novel cardiac therapies for the patients and families who need them. So today I'll be able to talk with you briefly, highlighting the evidence we have, the foundational science we have in disease models, specifically for the 2 clinical trials we're currently enrolling, which the cardiologist just discussed. So next slide, please. So starting with TN-201, in order to test for the first time whether AAV, restoring functional copy of myosin-binding protein, could help to improve the heart health of symptomatic mice with the mutant myosin-binding protein gene, we generated our own in-house model, which you can see here in blue. Now, this model, as you can see, starting on the left, exhibits severe hypertrophy. So if you look at the leftmost chart, you can see this model lacking myosin-binding protein in blue has a severe increase in heart mass compared to the healthy control in gray. Additionally, there's decreased heart function, as you can see in the middle graph in blue. And these mice as well have decreased survival. Now, to be able to demonstrate that we were able to generate a therapeutic benefit in this mice, a single one-time administration of TN-201 to these 6 mice, as you can see, decreased the heart mass. That's demonstrated in orange on the left. It also increased heart function, as you can see, comparing orange and blue in the middle graph, and resulted in a normal lifespan in these mice, as you can see on the right. Next slide, please. Now, very similar to what we did for TN-201, for TN-401, we utilized a model in which the mice are lacking functional PKP2. And what you can see is that these mice exhibit severe arrhythmia, and that's indicated in the second box, compared to the healthy animals with the normal rhythm in the top box. Now, if you provide a one-time injection of TN-401, you are able to prevent the emergence of these unhealthy rhythms, as you can see in the third box. So very similar to TN-201, with TN-401, you're able to restore normal levels of the missing protein and improve survival as well. And now at this point, I think we'll transition to answering any questions you guys might have. Thank you.
Whoop. Trying to come back on camera. There we go. So thank you. I want to first thank all the speakers for coming and sharing your insights with us this evening. Please continue to add your questions into the Q&A feature. If we can go to the next slide quickly. So we will leave this slide up while questions are being answered, and the folks in the background will also be pasting these links into the chat function. So you will see these and we'll be able to click over to these websites and this additional information as we go through. And now I will turn things over to Michelle, who will help us moderate the questions. Thanks so much, Michelle.
Absolutely. Thank you so much, Wendy. So I'm Michelle Corral, and I head up corporate communications and investor relations for Tenaya Therapeutics. In my role, I help our teams field lots of questions from external providers. And so our group today asked that I help sort of quarterback the Q&A session and make sure that we can get through as many of your questions as possible in the time allowed, since that truly was our purpose in this event. So I see a few questions have come in through Q&A. I'll do my best to try and group them kind of by topic. I think the first couple that I saw come up were really more related to TN-401. And so I'll kick that off, and I'll ask our speakers to also join me on camera to be ready to answer as well. So I think that one of the first questions that came up was just how many ARVC patients have received gene therapy so far? And have any of them had adverse reactions? That feels like something in your court.
Yeah, I mean, at this time, we're not to the point where we're able to discuss the actual dosing of individual patients. We certainly look forward to that time when we are able to discuss that. But again, at this point in time, we're just not able to discuss that.
Yeah, I would add that I know there are a couple of other sponsors who are studying gene therapies for the potential treatment of ARVC. And we have not, like from where we sit in terms of sort of monitoring, we have not had anybody announce a first patient dosed. But that doesn't mean it hasn't happened. I think it's up to the company's discretion likely as to when they would choose to make such an announcement. Another thing that we could add is, it is a clear sign of like a very severe adverse reaction, and we could probably talk to this more, would be a clinical hold situation. We haven't been hearing anything about anything like that either. That doesn't mean that nobody's having any adverse reactions whatsoever, but nothing that the FDA needs to more closely examine has occurred to date. Another question is about our gene therapy. And, I think going back to some of the data that Laura was sharing as well, does our gene therapy hope to remodel the heart to correct damage done to it by ARVC? And I would say we should also think of that question in regards to HCM as well.
Yeah, absolutely. That is our hope, and that is certainly what we've seen preclinically, is that with the restoration of normal levels of the functional protein over time, the heart can be remodeled, or you have basically a preservation without further decline.
Great. Thank you, Laura, for jumping in there. And then I think another question that applies to both, and with this feels like one for you. Will medical insurance eventually pay for gene therapy?
Yes, that's the usual situation with the approved gene therapies, that there is coverage provided. Some countries will cover gene therapies, but private insurance will as well. Obviously, there's a lot of discussion between providers and insurance companies, but the vast majority of gene therapy payments are made by insurance companies.
That's awesome. I have a couple of new questions regarding MyPPC3 HCM and our MyPeak study. One of those is, and this is a question that I'm fielding quite a bit, actually, is when will the data from the phase 1 trials be available? And so I can tell you that Tenaya plans to report initial data from this ongoing phase 1b study of TN-201 in the second half of this year. So those data will focus on safety. We're also doing heart biopsies to make sure that gene therapy is actually reaching those cardiomyocytes, the special cells in the heart where protein is produced. And so we want to make sure that the drug is getting there and see where, are those little cellular factories starting to get up and running. And then we'll also likely be able to look at biomarkers that are one measure, and which should explain this, of heart health. And that'll be for the first few patients that we've enrolled. Since this is the first gene therapy that has been studied in MYBPC3 HCM, there's going to be a lot that we're still learning, and we're collecting tons and tons of data. But this first data readout is really focused on that safety and evidence that we're getting into target cells. And then once we are able to confirm that that's happening, we hope that the results just continue to improve over time, and we learn more about dosing and so forth as we continue testing. Whit, let me know if I missed anything.
No, great job. We very much look forward to sharing initial data. As Michelle said, this is initial data. We do not anticipate that it would be conclusive regarding the effects of gene therapy or the safety of gene therapy, but it will be an initial understanding of how well it's working. So we very much look forward to discussing those results. And as Michelle mentioned, there are many different ways of looking at how it's working that we'll be analyzing, and some will be early and that we can discuss as early as this year, and others will be mature at a slower time course, and we'll talk more about those next year.
Yeah, and I think that kind of leads into the follow-up question there, which is, whether or not, our initial study in MYBPC3 HCM or potentially even the preclinical evidence, do we think that gene therapy could result in improvements in any of the secondary diseases that can occur as a result of HCM?
By interrupting the disease process of HCM, we certainly hope that gene therapy will prevent the cascading events, for example, that can lead to end-stage heart failure.
Awesome. And then while we're still kind of on the subject of clinical trials, I know you talked about the investigational nature of the drugs and things that could be of safety concerns. Can you talk a little bit, you and Matt, about what Tenaya has put in place to kind of ensure and monitor patient safety?
Patient safety, participant safety is our top priority, and that is really how the protocol is designed to maximize our ability to monitor safety and react quickly if needed. So the visit schedule and the assessments at those visits are all designed based on what's already known about AAV-based gene therapies. As I showed, there have been multiple approvals, far more clinical trials for many different types of gene therapy. So our understanding of what happens when in the body's response to a gene therapy is well fleshed out. The FDA has had joint meetings between sponsors, patients, and academic researchers to all align on these. So we have a lot of data. We had a lot of data to leverage when we designed these protocols.
Another question that I think is a really good one, and this may pertain, let's first do to 201 and then 401, but what are some of the criteria for being able to enroll in one of Tenaya's gene therapy studies?
Yeah, I can answer that. And I also encourage individuals to use the resources. Certainly what's posted on Clinicaltrials.gov is very informative and should be able to provide a fair amount of information. At a high level, of course, you need to have an appropriate mutation that causes a lack of a functioning protein so that you're in what we efficiency. We talked about the need to have some evidence of some signs and symptoms of the disease. So some evidence that your heart muscle is under some level of strain and that there's some abnormality there. Certainly you want to have some level of thickness so that you do have that diagnosis of hypertrophic cardiomyopathy. And again, in this initial phase 1b trial, so the initial look at the safety primarily of the TN-201 gene therapy, we want to make sure that you do have an ICD for this initial study. Does that mean that this will be the criteria in all studies going forward? Well, certainly that's not the plan. And we would hope to be able to broaden the opportunity for individuals with a different profile and broader profile to be able to participate. But for now, of course, as in all trials of this type for these very investigative and early therapies, the criteria are much more limiting and limited than they will be in subsequent trials.
And then, Matt, I apologize if I missed it because I was actually reading the next question. But did you mention as some of the enrollment criteria for TN-401 and ARVC?
Yes, again, I encourage you to go to Clinicaltrials.gov. But I think we covered kind of the high-level criteria of needing to have the appropriate pathogenic or likely pathogenic gene mutation, some evidence that you're at risk for arrhythmias, and the best risk indicator that's measurable is the number of PVCs or irregular heartbeats that you have over a 24-hour period. You do need to have a functioning ICD. Certainly that's an important readout for the trial and safety for those participating, because there was a reason why they had the ICD implanted. And then there is an opportunity for individuals, whether you've had a VT ablation or not. And if you've not had a VT ablation, then that's of course fine. And if you have had a VT ablation, as long as you're 6 months removed from that procedure, you would be eligible for the study.
Awesome. Thank you for that. Kind of on that topic, jumping back maybe to safety, we just had a question about, kind of asking, so what, like we talked a little bit about high level risk, but this question was a little bit more specific. What might the worst case scenario be in terms of gene therapy side effects? Is there something really, I think just a better understanding of what are some of the risks. I know we talked about, sort of the close and careful safety monitoring and mitigation plans that we've put in place to ensure that that cannot happen. But just explaining a little bit more about, what is it that people need to be the most cautious of and thinking about, sort of the weighing the pluses and minuses?
Sorry,
Maybe.
sorry. Who's that question for?
I'm going to give that one to Whit because I think that's kind of a big picture of how
Sure.
gene therapy works.
Yeah, so as I mentioned, what's been seen for the approved gene therapies and in other clinical trials is that the immune system's reaction to the delivery method, that viral vector, is what we need to watch for most. The other types of concerns that can happen with a gene therapy are less relevant for our particular forms of gene therapy. So, for example, in Duchenne's disease, muscular dystrophy, and other diseases where the patients have never seen that protein before, the immune system believes that protein is foreign and attacks it as well. That doesn't happen for either MyBPC3 HCM or PKP2, or it shouldn't happen. We don't expect it to happen because patients already have the protein. Only one of the two copies of the gene is mutated. So we do not anticipate any immune reaction to the therapeutic protein. It's just about the delivery capsid, which is only there temporarily. So the immune reaction most commonly takes one of two forms. One is inflammation in the liver, and the other is in blood proteins called complements. In both cases, we have very good blood tests, and we can monitor very quickly, or in real time, essentially. And in both cases, we have reactive responses, medications we can use to stop that type of reaction early. So the biggest risk is if a participant misses visits. So compliance in the trial, getting those safety blood tests at the appropriate time is really the most important thing.
That was a great question, because I think that was Whit, thank you. That was a really informative answer. And then the next question I think that we have is, sort of looking ahead. On each of these trials, the question is kind of, is there a timeline for a phase 2 trial commencing? I assume that is related to MyPeak. And Whit, I know we kind of went through like just the importance and the incredible value the patients bring to the entire clinical trial process, and said that there wasn't quite time to go through what the different phases are, but maybe we make a quick exception and you can explain a little bit of that phase 1, 2, 3, and how it's a bit different in gene therapy.
Yes, happy to do that. So a traditional often for small molecule pill type therapies process has very clear phase 1, phase 2, phase 3 clinical trials. Phase 1 is about the safety and whether the drug is having a biologic effect and getting into the bloodstream at the appropriate amount. Phase 2 is the initial proof of concept and the dose ranging. So initial evidence for efficacy and then picking the dose that has the best efficacy profile and safety profile. And then phase 3 or pivotal is pivotal because those are the studies designed to convince the world that it's safe and effective. And the world includes regulators such as the FDA. So that's what leads to approval, where the drug can then be used in regular clinical practice. And then phase 4 is additional studies to learn more about the drug or in new indications, new diseases after that. Now, in orphan diseases and in gene therapies, often more than one objective or more than one phase can be combined in a single trial. So in both cases, both of our gene therapies, we are in the first in human trial, which means safety, safety, safety is the first, second, and third goal. But we are also achieving phase 2 type objectives, getting an initial evidence of efficacy and identifying the best dose to take forward into the pivotal trials. So in many cases in gene therapy, the second trial is the pivotal trial. I will say that this is something that Tenaya and other sponsors work closely with the FDA. The FDA is an important partner and a very good partner, and very engaged in making the development process as scientifically rigorous but as efficient and fast as possible, because they recognize patients need new options now, and so they're trying to facilitate drug development. And you can look online for for Peter Marks at the FDA, who's really behind a real push to respond to patients and to bring forward gene therapies efficiently to get them to patients who need them. So I can't give specific timelines. The data will tell us when it's appropriate to move to the next step. And we look forward to working with the FDA as we define what our next steps will be.
Awesome. And then Matt, I had one for you. Could you talk a little bit about what participants might be able to expect during a clinical trial in terms of sort of the types of assessments and time commitments and just what's the flow?
Sure. Yeah, as you can imagine, as Dr. Tingley outlined, job 1, 2, and 3 is about safety, particularly in these early experiences. And that's our job. And certainly the FDA is our partner in helping craft that and making sure that we have the appropriate types and numbers of assessments to ensure patient safety throughout the entire trial. Obviously, it's most intense within the first several weeks after dosing, predominantly focused on safety-based assessments. And there are, quite a few. But again, it's all about ensuring the safety, understanding how each individual's body may be reacting to the introduction of the gene therapy. And as Dr. Tingley explained, it's really the reaction to the virus and the capsid. And we want to be able to monitor and control that appropriately and to get you through that most vulnerable period. As time progresses, yes, we will continue to monitor signals regarding safety, but we'll also be looking at potential evidence of how is this affecting the disease state itself. So how are some of the markers of the severity of disease, whether the markers are in the blood, whether that's evidence of some changes in structure and function of the heart, predominantly measured by echo. And then other measures, of course, that are tailored to the disease. In the case of hypertrophic cardiomyopathy, do we see any change in your ability to function and to exercise? In the case of ARVC, do we see reduction in the risk conferred by electrical instability as measured by PVCs? Do we see that change over time? So, all of these assessments are, again, most intense in the first several weeks to months, and then over time, the assessments become less burdensome for individuals. And the first year is the most intense. After the first year, the FDA does require that we do continue to follow individuals over the next several years, up to 5 years. But again, the assessments there are largely driven by safety check-ins, and again, much fewer of those in years 2 through 5.
And then I think another important question that's come up is, if you're participating in a gene therapy clinical trial, and you have one of these conditions and you're symptomatic, and so you're probably likely already on some form of existing treatment. Are you able to discontinue your medications or have your ICD removed, for instance? How does that work? And that's
Sure.
again for Matt.
Sure. And these obviously are early stage, and we want to make sure that we keep safety in mind. So it's the best starting point is to make sure you're on the highest standard of care that's most appropriate for where you are in your disease spectrum, and that you would continue that. Any changes to medications should really be in consultation between you and the study doctor, and probably more important, your caring cardiologist in each of these cases. What would the ideal scenario for all of us involved in this be that individuals eventually in the future are able to reduce medications and perhaps, again, I don't know that we necessarily take an ICD out depending on the type of ICD, but maybe not have to implant them or certainly not replace the generator when that's time. I think that's a future goal, but we got to take this one day at a time and one patient at a time and see that we can get there. But that would be the hope for the future.
Cool. And one more question related to clinical trial participation, and then I have one that's going to involve early research coming up. So I just wanted to give Laura a heads up. So with regards to, as we're all kind of anxiously awaiting the ability to share data, can we expect to hear from clinical trial participants, or do they have limitations on what like an NDA or anything like that in place? How does that work? And I think Matt, either one of you can take that. Both experts on this topic.
Yeah, I think what's going on.
Patients are not required to sign non-disclosure agreements or NDAs to participate in our trials. Freedom of speech is freedom of speech. However, we do ask, and investigators, when you talk to the study doctor, they will probably ask that you choose to refrain for the integrity of the trial. So there are a variety of reasons why initial discussions by participants could affect the interpretation of the overall trial. It affects how future patients might think about their experiences in the trial. So it creates bias that way. Or, in the case of, well, every patient is different. But the first patients could set expectations for future patients, which also introduces bias. So it is better for the scientific integrity of the trial if participants aren't directly aware of the experiences of the other participants. So that's why your study doctor will recommend that you not post on social media and do those sorts of things. So, of course, talking with your family, your close friends is different, but in forums where there are other participants, there's a preference that you not share.
And I can add, having worked at other companies that are developing all kinds of medicines for different indications, whether that be cancer or asthma, that is a very common sort of policy, because nobody who's doing this kind of research wants to put those clinical trials in jeopardy and have a result that's not truly scientifically based. Speaking of science, thank you, Laura, for popping back on. We have one last question that I wanted to be sure that we addressed about sort of the potential for research in other gene mutations. Our asker had a very specific one in mind. I don't know if we want to talk to that specific one, but I'd love for you to tell us a little bit about what we can share about Tenaya's early research and kind of our philosophy.
Yeah, absolutely. Thanks, Michelle. And we're right at time, but I will say, absolutely, we always have, following the human genetics, following the patient need, we always have many early research projects in the pipeline. And the ones I was able to talk to you about today are obviously ones that we're in clinic with starting now, but we always have many in the pipeline that are not just gene replacement, but also gene editing in different indications, such as, TPM, et cetera, as a potential. So yeah, we're always looking at new opportunities to help patients. So thank you for the forward-looking statement, and hopefully you'll hear exciting news from us on other trials coming soon.
Cool. Amazingly, I think we got through the questions that we received. Thank you to all of you who tuned in and participated in such a way so that I wasn't just standing here. And I'm going to turn it back over to Wendy.
Yeah, thank you so much to everybody, to all the speakers tonight and all the people who attended. I hope you take advantage of these resources that we've provided here, and certainly please reach out to patient.advocacy@tenayathera.com if you have further questions, things that just pop up at another time. Family asked questions after this, and we did record this session, and we will be posting it to our YouTube channel. Don't know exactly how quickly that will happen, but we will certainly let people who registered for this event know when that happens, and we'll make sure to get that out there. Also, I want to just put in a really quick plug. The Patient Advocacy Department at Tenaya Therapeutics has now started a newsletter. So if you would like to be on our newsletter list, just send a message again to patient.advocacy@tenayathera.com, and we will make sure to add you so you will get those newsletters. So thank you so much to everybody. It was a great presentation, and thank you for our audience, speakers, everybody, and have a lovely end to your evening. Bye, everybody.